we're very excited by this first-in-human study. CC-92480, as you very nicely described it, I'll call it 480 for short, is really very impressive small molecule. It's a potent novel cereblon E3 ligase modulator, hence the term CELMoD, and it's specifically designed for the rapid and maximal degradation of target proteins, and in particular here, of course, Ikaros and Aiolos. Now, this has preclinically shown remarkable direct anti-myeloma and immune stimulatory activities in preclinical models. And this has been very striking in even lenalidomide and pomalidomide resistant myeloma preclinically. And moreover, when you combine 480 with standard of care therapies such as dexamethasone, proteasome inhibitors, and monoclonal antibody therapy, there's clear synergy preclinically. So there's a very strong rationale for this drug. And it's important to note that it was a designed molecule, and it was designed, as I say, to not only rapidly but maximally degrade the target proteins. So this was a large multi-center international phase 1, so really not really quite an ambitious early phase trial, and it was a privilege to work with an outstanding team, actually. And what we were able to do is we've been informed by really nice preclinical work about the importance of either exploring a continuous schedule or an intensive schedule. And to cut a long story short, using basically an adaptive design with a Bayesian model, we identified the MTD at 1 milligram per day and the best schedule as 3 weeks on and 1 week off. What was particularly elegant about the trial was the correlative science. The correlative science was dynamic and patient-derived. We did this by carefully sampling correlative science based on pharmacokinetics and pharmacodynamics. And we did this by looking at Aiolos degradation, and we did this by a very dynamic approach to response assessment, which allowed us to demonstrate that you needed continuous treatment for 3 weeks on and 1 week off to maintain response, but also that the 1 milligram dose was optimal at maximum Aiolos degradation. And this was sort of really important. And we also then correlated it with myelosuppression. And, how long did you need for count recovery? And we were able to demonstrate very convincingly then that the 3 weeks on, 1 week off schedule was what you needed. Now, the data we presented was on 76 patients. So it still remains preliminary because we've completed the first phase of the study and we're now expanding in the RP2D2 dose, or recommended phase 2 dose. And of the 76 patients, these were all relapsed and refractory. And what was the requirement at each of the centers is that every patient had to have exhausted the available approved options at that particular center. So everyone was basically proteasome inhibitor refractory, IMiD refractory by virtue of POM and lenalidomide exposure, and monoclonal antibody exposed. So the true sort of classic triple class refractory group that were penta refractory was actually half the population, which in a phase 1 is pretty strong. And a third of the patients had extramedullary disease. And as I've mentioned, all of them were relapsed and refractory, and almost everyone had been triple class exposed. So this was a really vulnerable population. And we demonstrated clearly that myelosuppression was the most important side effect, but was manageable with dose reduction, GCSF support, and so on. We didn't have any treatment-related mortality, which is really nice. 76 patients strong, that's quite a reassuring safety signal. And we saw very little in the way of sort of non-hematologic side effects that would be in any way concerning. There was only one case of deep vein thrombosis, for example. Peripheral neuropathy occurred in less than 5, it was only in 4 patients, if my memory serves me rightly, and it was all low grade, grade 1, 2. And then very interestingly, we saw 2 cases of grade 2 pneumonitis that were manageable with supportive care and dose reduction, but nothing grade 3. The biggest challenge we saw was chest infection, but again, very consistent with this population. Around 22% of patients, and very importantly, it was manageable with appropriate antibiotic support and so on. And the rate of febrile neutropenia was also extremely low. So myelosuppression a major issue, but manageable. And at the recommended phase 2 dose, clearly we think we've got a platform that can go forward. Now, what about responses? Well, in terms of responses, taking the fact this is a true phase 1, if you look at the intent to treat population, the PRs or better were 21%. If you drill down at the recommended phase 2 dose group, actually it was 55%. And what was really striking is that 70% of these patients were triple-class refractory. And in that group, we had complete responses and VGPRs. Now, the numbers remain relatively small, so you have to be a bit careful, but I will say that was, to us, very encouraging. And what was then particularly interesting, Charlie, is we looked at the extramedullary group, because this is a big problem. And we had, as I mentioned, about a third of patients who had extramedullary disease. And at the recommended phase 2 dose, and importantly at 1 milligram, we saw striking activity against extramedullary disease. And I presented that at the session. And I think for everyone, that was particularly noteworthy. So right now, where are we going? Expanding the recommended phase 2 dose, and we're also combining 480 with other standard of care drugs going forward in another large trial. So that all being the case, we're very hopeful that this will be, building on the success of iberdomide, or CC-92480, and if I get my numbers right there, 92480, 480, it's almost like models of the Airbus, isn't it? 220, 480. The 220 is moving forward a little bit earlier. We're very much moving into this tough relapse refractory space, and we're very hopeful we'll see continued success for this molecule.